Hello everyone, I am Priyanka Arora from Department of Biotechnology, Rama University. I am here again to give another lecture on my molecular biology series. This is RNA interference, which is also known as RNA silencing. So, I am talking about RNA silencing or the RNA interference. It can happen with the presence of more than one type of RNA. There are many types of RNA present. One such type is miRNA, which is also known as microRNA. This microRNA is involved in silencing of the gene product. Silencing means the mRNA, which should be converted into the protein, is now blocked and not allowed to express itself. This process is known as silencing of the mRNA and not allowing it to form the protein. This process is known as RNA interference or RNA silencing. So, this can happen with the help of miRNA, which is also known as microRNA. This miRNA are encoded by the eukaryotic genes itself. There are around 1500 types of genes which code for miRNA. Okay. These miRNA are the non-coding genes. They do not code for any protein. These are also known as sTRNA. sTRNA, which stands for short temporal mRNA, short temporal RNA, which is also same as miRNA. Okay. So, these miRNA, these are involved in cell differentiation, where some of the genes are not required to be translated, so that there is a formation of different cells in different functions. Okay. So, for example, if we have a skin cell, so this is a skin cell and this is a nerve cell. Okay. The same mRNA, the same chromosomes are responsible, the same number of chromosomes, 40 chromosomes, 46 chromosomes are present in this cell also and 46 chromosomes are present in this cell also, but one cell is responsible for the formation of the skin cell and the other cell is responsible for the formation of the neuron. How is this happening? This process is known as cell differentiation and it requires many type of post-transcriptional modifications. One such modification is miRNA, that is not allowing the certain proteins to be translated and allowing different types of formation to happen, or it is also involved in cell death, apoptosis and various other cell proliferation mechanisms. Okay. Let us talk about the process, how this process happens. Okay. So, we have it starts in the nucleus. So, in the nucleus we have a gene, the chromosome, which codes for some genes. The genes involved in these chromosomes are responsible for the formation of miRNA. Okay. So, the gene suppose codes for the miRNA. This miRNA, when encoded or translated, after the transcription forms the mRNA. This mRNA has a complementary sequence present in the nucleus, which comes and binds to this miRNA. Okay, and it forms a imperfect hairpin loop. This imperfect hairpin loop of the miRNA is also known as preliminary or pre-miRNA. This is not the mature miRNA, this is the preliminary miRNA, which needs to be modified to form a mature miRNA, which is responsible for the silencing of the gene. Okay. This miRNA is now required to be modified. This modification is done with the help of various endonucleases. Okay. So, if we talk about nucleases, we know that these endonucleases are responsible to break the nucleotide bonds. It will break the nucleotide bonds. Okay. So, one such endonuclease is DROSHA, which comes and binds, and it will help in for changing or modifying the structure of pre-miRNA. This pre-miRNA is around 1000 nucleotide long. After its modification, with the help of the DROSHA enzymes or the DROSHA endonucleases, it gets converted into a much smaller or the shortened structure, which is known as pre miRNA, also known as precursor miRNA. This precursor miRNA is just 70 base pairs long or 70 nucleotide long. Okay. So, this happens with the help of the enzyme or the endonuclease drosha. This drosha requires the help of another enzymes which help in loading the drosha onto the pre-miRNA. Such enzymes are known as RNA binding proteins. This RNA binding proteins, example is PASHA. This PASHA is the enzyme which is the RNA binding protein which helps drosha loading onto the pre-miRNA. Okay, but this PASHA is present in Drosophila. And in mammals it is known as DGC8. Okay, this enzyme is known as DGC8. Okay. So, now after this, all this thing is happening inside the nucleus. Okay. After this pre-miRNA, it requires another modification, more sets of modification to become mature miRNA. For that, this requires to be exported outside the nucleus and reach the cytoplasm where the process of translation happens. Okay. So, this miRNA, if it requires to go out of the nucleus or the nuclear pore, it has to have another set of proteins that will help in export of it. So, the export protein here is known as exportin 8. Exportin protein. Okay. So, this exportin is required for the transport of pre-miRNA onto the cytoplasm, and the next set of modifications happens in the cytoplasm. Okay. So, now let us continue. With the help of the exportin in the cytoplasm, what happens? With the help of exportin protein, exportin 5, pre-miRNA is transported into the cytoplasm. Okay. Here in the cytoplasm, more sets of modifications happen for it to become a perfect miRNA. Okay. So, how it becomes? Now it requires another set of endonucleases. These endonucleases which are specific only for the cytoplasm, and these are known as dicer. Okay. So, dicer are the endonucleases which acts upon pre-miRNA, which is just 70 nucleotides long, and it will further modify these pre-miRNA. It changes its structure for it to become perfect miRNA. So, the structure was this, the imperfect hairpin loop. Now, when it is acted upon by the endonucleases, it cuts and it forms miRNA duplex. This is miRNA duplex. This miRNA duplex is now further shortened, and it is 22 base pair long only. Okay. So, this shortened miRNA duplex is now ready to attack the miRNA which is to be degraded or not allowed to be translated. Okay. So, how it happens? This miRNA is required to be degraded further such that it is only left with single miRNA. Since in the starting I told you about the miRNA which was present in the cytoplasm, which was present in the nucleus, so that it comes and binds to the mRNA which was formed of the dicer. Okay. Now, that miRNA has to be released. So, this miRNA is known as passenger strand, and this strand of the miRNA is now known as the guide strand. This is known as the guide strand because it has a sequences which will allow us to guide the mRNA which is to be degraded, and this is a passenger strand because this has allowed the mRNA to come out of the nucleus, and it will now it is ready to be degraded. Now it is not of any use, the passenger strand is not of any use, so now it has to be degraded. For that degradation process, there is another set of enzyme or a box, a complex of enzyme which is known as RISC box. Okay. This RISC comes with the help of Argonaute protein. Okay. This Argonaute protein and RISC is known as RNA induced silencing complex, RNA induced silencing complex. Okay. This RISC with the help of the Argonaute protein, it binds to miRNA duplex. Okay. This Argonaute is responsible for the degradation of the passenger mRNA. It degrades the passenger mRNA and allows a miRNA to be free and now ready to find its target. Okay. So, now we will look at its target here. So, its target could be any mRNA whose complementary sequences are present in this mRNA. Okay. So, this is 5' end of the mRNA and this is 3' end of the mRNA. Okay. At the 3' UTR region, this is untranslated region, that is, it is not coding for any protein. This untranslated region has a sequences which is complementary to miRNA which is now created. Okay. If the signal comes that this mRNA needs to be degraded, this miRNA will be produced and this miRNA will go and bind to the untranslated region of the mRNA which is to be silenced. Okay. And it forms a complementary strand to it. Okay. So, after this attached, this is my miRNA which is a guide miRNA, and this is my mRNA which is to be silenced. Okay. How it silences? It combines two processes. It can happen in two different ways. In first way, it comes along with the protein RISC complex as well as Argonaute. Okay. So, this Argonaute and RISC complex can degrade the mRNA by mRNA degradation. What happens in this Argonaute and RISC complex? It degrades the mRNA to which it is bound. Okay. And thus it will not allow the translation to occur because there is no mRNA for the translation to occur. Okay. In the other process, what will happen? This is the first process, or in the second process what will happen? It will block the ribosome from coming and binding to the mRNA, and since ribosome is very essential for the formation of the protein or for the translation process to happen, it will not allow the translation to happen and thus it will block the translation process. Okay. So, it will not allow ribosome to come and bind. Okay. It will not allow the ribosome to come and bind to the mRNA. Okay. Thus, it is blocking the translation. So, in this way mRNA which is to be degraded, which is to be silenced or not allowed to form a protein in a particular amount of cell, in the particular cells have the mRNA which is complementary to their UTR regions present inside them. Okay. In this way they are allowing it to be silenced. This technique is used in various laboratory purposes also to degrade a specific mRNA. In this way we will degrade or not allow a certain protein to be translated in a cell by forming a complementary miRNA outside the cell and inserting inside the cell, and thus we can stop the translation process. Okay. Similarly, RNA interferences are also happening with the help of other mRNAs, other RNAs also, and one such RNA and one such technique is siRNA, or this is also known as, one such technique is, suppose this is second because this is the first we are talking about. Okay. This siRNA is known as small interfering RNA. Okay. With this I finish my lecture. I hope the mRNA technique is very much clear to you. Thank you.